Let's welcome Gil.
Well, hello everyone, and thank you very much for making it here so early on a Sunday morning. I'm sure that the top on our list of things to do on a Sunday was to be in a hotel auditorium, so I really thank you for making the effort to be here. And since I'm the first speaker up, I do promise to make it concise. I'll make it to the point, and I hope that what I'm going to speak about today is interesting, and hopefully it's worth your time. So I joined A5M a little bit earlier this year, soon after turning 20. And you're probably asking yourself, what on earth is a 20-year-old kid doing at an anti-aging conference? After all, I'm probably the spring chicken in this room. I should be using my ample testosterone, my ravishing good looks, and awe-inspiring muscle mass outside. But instead, here I am in the Sofitel auditorium of the 7th annual conference in anti-aging medicine. And the reason I'm here is because I'm contemplating what I'll look like at age 40 if I don't take the advice of the wonderful speakers that we have at this conference. Now, I come from a family which has essentially been doing homebrew anti-aging for a number of years now. And back in my hormonally optimized days as an 18-year-old man, many years ago, I used to be very skeptical about this whole idea of anti-aging, the anti-aging phenomenon. What does it mean? But then I started to see the effects on my mother and father, and when friends started making comments that I have a yummy mummy or a MILF for a mum, then I knew anti-aging is real, it's working, and I can't deny it. So I'm no longer skeptical. And now that I'm heading into the last 2 years of my medical degree, my fascination with the topic of anti-aging has only grown further. And being at a conference like this is a really wonderful experience, and I'd hope to collaborate with some of you in the future. So working together, we can hopefully look as beautiful as we do today for many more years to come. So today I'd like to discuss a literal blind spot of aging, an elephant in the room. Believe it or not, it's our eyes. So emerging research suggests that as we age, functional and anatomical changes in the human eye can radically affect the generalized human aging process. Aging of the human eyes can actually result in accelerated human aging. Now, it seems sort of strange that something as seemingly trivial as worsening eyesight, worsening vision over time, can have such a dramatic effect on our health. So if you can't read what's on this slide at the moment, you might be in a little bit of trouble. Okay, so recent studies looking at how the eye ages have given rise to really incredible technologies in the fields of medicine and neuroscience. And this is particularly so with regards to stem cell kits. So with these advances, I'm telling you that some clinically blind people may be able to see. We may have new emerging therapies for Alzheimer's and Parkinson's disease that can be used as an adjunct to current therapies. And a 50-year-old you may be able to feel like a 20-year-old you again. Now, you're probably thinking, what is all this research I'm talking about, and why on earth, of all things at an anti-aging conference, should I care about the eyes? So first, let's take a look at what can go wrong as our eyes age. We can end up with disruption of the body's internal clock, a complete disruption of our circadian rhythm, our day-night cycle, our sleep-wake cycle. We can end up developing chronic illnesses such as diabetes, obesity, cancer, hypertension, and hyperlipidemia. And perhaps most worrying, we can actually suffer an early mortality. We may actually be dying earlier as a result of our eyes aging and not addressing this sufficiently. So before we get into the specifics of the really interesting mechanisms underlying this, I just thought I'd take the opportunity to briefly outline some of the basic function and anatomy of the human eyes, and hopefully this will put some later parts of the talk into more perspective. So at its very basic level, the eye is a sensory organ. It permits our vision, and it also permits the physiological responses to the non-visual effects of light. So we'd probably all be familiar with the two main cells in the back of our eye, rods and cones. We've probably all heard of them. So rods and cones, they're the cells that allow us to distinguish over 10 million different colors. They give us a perception of depth. There's around 125 million of these rods, 6 to 7 million of the cones. And rods, they basically allow us to see in the dark, so they respond to low intensity of light, and some studies have recently indicated that as low as 1 photon of light is enough to elicit a response. In contrast, our cones are our vision in the light, so they function optimally in bright light, such as bright outdoor light. Now this is where it gets really interesting, because back in 2002, a whole new class of photosensitive cell was discovered. And this cell has been found to be involved in synchronizing our circadian rhythms. This particular cell type is the final output neuron of the retina towards the brain. And it is involved in the suppression and release of melatonin from the pineal gland of the brain. And it does this in concordance with the degree of light stimulus. Okay, and I thought I'd bring this image up because it's a macro scale of the eye, and we often hear terms like the cornea, the pupil, the lens, the iris, and the retina without really fully understanding at a basic level what that really means. So firstly, the cornea, which we can see as the front part of the eye, that's the transparent front of the eye. And adjacent to that, the sclera. The sclera is the white of our eye, and that's a collagen-containing protective layer. Now the lens we'd all be familiar with. The lens is a crystalline structure, and that refracts light onto the retina to be focused. We have the iris, which we'd all be familiar with as the coloured parts of our eye. It's what allows our eyes to appear green or blue or brown. And that structure controls the diameter of the pupil, so it's not just there to give us colour. And finally, probably the most interesting aspect of the macro structure of the eye is the retina. So the retina lines the inner surface of our eye. It's a light-sensitive tissue that responds to light stimulus, encodes this into an electrical signal, and sends it directly to the brain via the optic nerve. Now, not long ago, I just mentioned the term photosensitive retinal ganglion cells, and this is some really interesting emerging science. So I just wanted to briefly mention a little bit more about these. So these were discovered back in 2002, and although they are a photosensitive cell, they are novel and they are radically different to anything else that we have in our eyes. So they're a completely new, unique cell type. And they encode a very special protein known as melanopsin. Melanopsin is a light-sensitive G protein coupled receptor, and what's fascinating is that it's actually remarkably similar to invertebrate insect opsins, so a light-sensitive protein. It responds to blue light, so short wavelength light, and has a peak absorption at around 488 nanometers. Now, the discovery of a cell type like this, something in the back of our eye that is controlling our circadian rhythms, controlling the release of melatonin from the pineal gland, that alone is quite astounding. And it's really astounding when we look at it in a more clinical context. So I just want to bring up an example of a more clinical implication. So in 2007, it was shown that in profoundly blind individuals with rare diseases that wiped out their rods and cones, that they still maintained normal circadian rhythms, normal photoentrainment, their pupillary reflexes were normal, and the regulation of melatonin was still normal. Now this is really counterintuitive to everything that we'd probably think about the eyes. So in order to have circadian rhythm, in order for our brain to be able to perceive day and night, sleep and wake, we at some level need to have a perception of light to be encoded into an electrical signal for our brain to be able to decode. Yet in profoundly blind individuals, people with rod cone dystrophy, people with retinitis pigmentosa, really devastating disease that are making them completely, clinically, profoundly blind, they still have these normal circadian responses. Everything seems to be going fine. In fact, the authors concluded that the subjects had no sleep disorders. They had normal age-appropriate 24-hour sleep-wake patterns, and this was confirmed by a number of different quantitative assessments. Okay, so essentially what this is telling us is that the duplex vision theory of rods allowing us to see in the dark and cones allowing us to see in the light is not the be-all and end-all. We have a new cell here. It's radically different, and it has a number of important implications, which I'm going to talk about. Another clinical context, which is really interesting. If we look at a disease like retinal degeneration, What we find is that these photosensitive retinal ganglion cells, these novel, radically unique new cell types, they're maintained. So we have degeneration of the retinal tissue, yet somehow these cells are still fine. They're functional and they remain. So what if we could do something with these cells that could address blindness? What if we could encode these cells, change them, fix them, do something with them that can be a real solution to medical problems? So this has spurred a lot of research in three key fields. So the first one is optogenetics. That's a technique I'll talk about a little bit later in the talk, which combines techniques of optics and genetics. The other one is in prosthetic tissue development, so prosthetic development of the retina. And another key one is in the development of stem cell kits. So this article here was published last year, and it's talking about a real stem cell kit in order to treat the world's most common form of cause of blindness, glaucoma. Another example here from the MIT Technology Review. Company aims to cure blindness with optogenetics. This technique will retrain cells that typically don't respond to light. And this also was published last year, and this is really happening. What this is telling us is that clinically blind people may be able to see as a result of just this one cell type. And just another example here. So this is published by CBS News, also last year, and they're talking about a new prosthetic treatment which helps blind mice see. So researchers aim for human next. Now, these are just a few examples, but there have been hundreds of articles, lots of research papers published about this topic, and a lot of us probably unaware that there's so much research happening in terms of the aging of the human eye and potential applications of this. So now we get to the interesting bit, which looks at what actually happens to the eye, our human eyes, as we age. So if you get anything out of this talk, what I want you to remember is that as we age, our eyes, they receive less light. And I put up here a number of key anatomical changes because it's important to consider the anatomical changes which compound the aging of the human eye. So we have thickening of the cornea. We have a decrease in the size of our pupils. The ciliary muscles in our eyes become weaker. We end up with a loss of retinal cells, and we also end up with a number of different blood vessel changes. And of course, we have the three key pathologies associated with aging. Glaucoma, which is the world's leading cause of blindness. We have cataract, which is an opacification of the lens. And inevitably, the majority of us here in this room will, as we age, develop cataracts. And lastly, age-related macular degeneration. So that's a very topical issue at the moment, AMD. And essentially, the clinical implication of that is that we can lose our central vision if it isn't properly addressed. Okay. A little bit more about the implications of the aging eye, and when we look at what happens to the eyes as we age, these photosensitive retinal ganglion cells, I know I'm talking about them a lot, but they become increasingly important as we look at the physiological changes that occur. So these photosensitive ganglion cells, they relay light information to a special area within the hypothalamus of the brain, known as the suprachiasmatic nucleus, or the SCN for short. Now, the suprachiasmatic nucleus within the hypothalamus, it sends circadian signals to a number of important organs, most notably the heart, the liver, and the pancreas. And by doing so, it also directly influences the release and suppression of melatonin from the pineal gland. But the really interesting thing is that it's also indirectly influencing the release of other hormones within our brain, and this can have a really profound impact as we age. So the key point, less light as we age results in less signaling to the suprachiasmatic nucleus of the hypothalamus, meaning that our circadian rhythms aren't being regulated properly, or we may not even have a circadian rhythm being regulated at all. Now, so this is an image. This is showing a sagittal section of the brain. That middle curved section, that's called the corpus callosum, and that separates the right hemisphere of the brain from the left. Below that, we have the thalamus, and below that, we have the hypothalamus. So I've underlined in red where the suprachiasmatic nucleus is situated, and also relative to that, there's a line showing the location of the pineal gland. Okay. Okay. So let's take a look at the lifestyle connection here. So as we age, we spend more time indoors. And the key point here is that indoor light is muted. It has a lower lux and intensity than bright outdoor light. Now, as middle-aged adults, we're spending on average around 1 hour each day exposed to bright outdoor light. Now, if we then look statistically at the amount of time that an older adult is spending outside exposed to bright outdoor light, it's halved. It's only half an hour. And as I mentioned earlier, these age-related anatomical changes at the macro level and the development of pathologies compound this effect. Now, this is really beginning to take a hold and is beginning to be recognised as a really important implication in the human ageing process. So Dr. D.M. Burson, his team discovered these photosensitive retinal ganglion cells. And he has said, anything that affects the intensity of light or the wavelength can have important consequences for the synchronization of our circadian rhythm, and this can have effects on all types of physiological processes. Dr. Patricia Turner of the University of Kansas School of Medicine has said, in modern society, most of the time we live in a controlled environment under artificial lights. These lights are 1,000 to 10,000 times dimmer than sunlight, and they're in the wrong part of the spectrum. And she also went on to say that she believes that the effect of this is huge and that it's just now beginning to be recognised as a problem. So let's take a look at what happens if we couple the effect of lifestyle, the fact that we're spending less time outside, and let's couple that with what happens as we age. So let's take a look at a nicer example at age 45. So at age 45, the photoreceptors of the average adult are receiving just 50% of the light necessary to fully stimulate our circadian rhythm. Age 55, just 37%. Age 75, just 17%. Now, this next image is really nice because it demonstrates this concept in terms of a graph. So what we see here is a graph that demonstrates retinal illumination relative to a 10-year-old's eye. So if we look at the line demonstrating what a 10-year-old's eye is, we can see that it gives a value of 1 in terms of retinal illumination. If we consider 1 to be our ideal, a young eye that is behaving normally. Let's now take a look at age 95 years old. We have around 1/5 of the degree of retinal illumination that a 10-year-old eye does. Now you can imagine what sort of havoc this is causing on our circadian rhythms, the regulations of hormones within the brain, all of these physiological processes being affected by something as seemingly trivial, something that seems almost as irrelevant as the aging of our eyes. I'll skip this slide because I've already talked about it a little bit, just in the sake of time. Now I want to look at some important implications of what this means. So if we know that light is good for our eyes, if we aren't getting sufficient light, our physiological processes are being compromised. There's been a number of developments at the moment. One of the big developments is in terms of intraocular lenses. So as we age, we often develop a cataract, and some of us in the audience may have had an IOL or an intraocular lens put in. So the new technology now is to have an intraocular lens that filters blue light. And there's been improved patient outcomes in some studies. Another one is elderly light therapy, and this has been shown to be really important in elderly people which sometimes have dysregulation of circadian entrainment. And particularly in elderly people, it's valuable in terms of addressing sleep disturbances and depression. My two favourites in terms of the application here are Alzheimer's and Parkinson's disease. Now, what a number of studies have found is that in the Alzheimer's and Parkinson's disease, something as simple as light therapy can improve their outcomes really significantly. And this is the case especially with blue light therapy. Another one, cataract surgery. So this improves the degree of daytime sleepiness and insomnia in some studies, and a few studies have also showed improved reaction time. Okay. What I'd like to now talk about is a study which looked at light-induced melatonin suppression and it looked at the age-related reduction in the expression of melatonin after exposure to light. So with this red arrow, I've highlighted what they found, and essentially what they found was that an old eye, in response to light, is not going to have as high an expression of melatonin. Now on the surface, taking this at face value, it probably doesn't mean much to us. Okay, so we have less melatonin, so what? Now, Dr. Patricia Turner, who I mentioned earlier, in commenting on this article, has said, what this shows us is that the same amount of light that makes a young person sit up in the morning, feel awake, have better memory retention, and be in a better mood has no effect on older people. And I'm sure we now have a much better appreciation of light. So circadian rhythms, what can go wrong when the circadian clock stops ticking properly? We can have fatigue, insomnia, weight gain, obesity, diabetes, cancer, psychological effects like depression and anxiety and memory loss. And, of course, we can end up dying earlier than we should. We can suffer an early mortality because we're not addressing in a sufficient way how our eyes are aging. Now, to finish up on something slightly differently, I'd like to talk about my research, which I did a few years ago, which looked at myopia, short-sightedness. So we already know that exposure to light and retinal signaling pathways have an important effect upon regulating our circadian rhythmicity, and also, very likely, have an important effect upon the generalized human aging process. So essentially what we did is we made these chickens, they were we-called chickens, we made them blind. We didn't make them fully blind, but we made them blind sufficiently that they developed a high degree of myopia. And then we wanted to see, if we looked at certain neurotransmitter interactions in the retinal tissue, could giving something like dopamine reverse or limit this process? If we gave another neurotransmitter like gamma-aminobutyric acid, GABA, what effect would this actually have? And what we ended up finding was that neurotransmitter pathways in the retina interact. This is a really complex interplay, and we probably don't give a lot of thought to the eyes, but the retina is almost like a little universe in itself. It's an entire nervous system. And what's fascinating is that so little is actually known about how the neurotransmitter pathways interact, what effect they have. And it's really important to think about, because the eyes, they connect directly to the brain by the optic nerve. If things change at the level of the eye, they will change dramatically at the level of the brain. And what we found was, in addition to the fact that neurotransmitters interact in the retina, if we expose the chicken's eyes to light, it can limit or reverse the process of myopia. Now, epidemiologically, myopia is a serious problem. The World Health Organization has identified short-sightedness or myopia as one of the world's leading causes of blindness. By 2020, over 2.2 billion people will be short-sightedness, and it's on the rise all over the world, even in Australia, and particularly among school-aged children. So this study is a really interesting one. It's called the Sydney Adolescent and Vascular Eye Study. Now what they found is that in kids that spent more time outdoors, exposed to bright light, they had a significantly reduced incidence of myopia. So exposure to outdoor light is inversely correlated with the development of a significant ocular pathology. So you're probably asking yourself, okay, so we know a little bit about the neurotransmitters in the eye, we have a lot more to learn. We know that light is good for the eye. So where does the future lie in terms of treatment? What would it take so that we no longer need spectacles like I'm wearing or contact lenses? And at the moment there's no simple answer. All I can really tell you is that we need to do more genetic studies. We need to knock in a gene that, let's say, upregulates the activity of dopamine. We know that dopamine is good for the eyes. What effect will that have? We need to knock out a gene. If we were to say, knock out the gene in the retina that coded for dopamine, what effect would that have? There's a number of other different techniques, such as transgenic studies, and a really fascinating one at the moment is looking at siRNA, so short interfering RNA, and that's a really emerging field at the moment. And I just wanted to mention that this has already been shown to work in rat models. So what is the light at the end of the tunnel? So already we have huge advances in the scientific and medical field in terms of understanding how the eyes age and what implications this has on the generalized human aging process. there's remarkable applications of this in terms of developing specialized medical technologies like intraocular lenses, light therapies, targeted light therapies to improve patient outcomes for people with Alzheimer's or Parkinson's disease. But more research is still necessary to elucidate the cellular and molecular mechanisms underlying aging of the human eye. If we are in the future going to have a targeted pharmacological intervention, not only to address ocular pathologies like glaucoma, age-related macular degeneration and myopia, but also to address some of the processes of aging in the human eye that impact on our overall generalized human aging, we need to understand this at the molecular level. Now, our understanding of the eyes is improving, and it's already giving us an indication how, when our eyes age, we can end up with chronic illnesses, dysregulation of our circadian rhythms, and possibly even suffer an early mortality. Now, just briefly, because this is a really fascinating topic, I want to briefly discuss the emerging field of optogenetics. So, opto and genetics. Opto, you take techniques from optics. Genetics, you take techniques from genetics. At the very basic level, optogenetics is a neuromodulation technique. You can change the activities of live neuronal tissue and modify the effects of this in real time. Now, this has really significant implications, and essentially what it allows us to do is use light as a switch. So we can encode a neuron, a neural tissue, to respond to light and work almost mechanistically as a machine. We can program our neural tissue. We can basically insert a genetic software into a cell that allows it to carry out a particular function of interest. And this is really interesting in terms of applications for blindness, epilepsy, and at the moment a key field of research is looking at the neurological disorders like things like schizophrenia. In terms of its implications for aging, who knows? And in the future, would we be able to download and upload memory? If we can already encode specific neuronal behaviors just using light, could the future mean that we would essentially have a light-activated, what's a way to put this? When we put a USB in our computer, we're downloading information to a hard drive. If our brains are the hard drive and we had some way of encoding information, or even allowing our brains to process at a faster rate, this would have immense implications. So briefly, how does optogenetics work? So we take a gene, a gene that encodes a light-sensitive protein. So we take the gene for that and we put it into the genetic code of neuronal tissue. Now, neurons, they interact by firing. They open channels which release ions. So basically our brain is swimming in a sea of electricity. Now, if we fire the right light on a particular neuron, we can get it to behave in a certain way. And probably the most fascinating aspect of this is looking at reactivating blind eyes. And just to finish up, I get back to this point of the photosensitive retinal ganglion cells that I mentioned earlier. We have clinically blind people that in all levels of typical understanding aren't able to see. They can't perceive light. They are clinically and profoundly blind. Their rods and cones are gone. What are we going to do to help them? These cells still remain. They are functional and they are programmable. So what's being done at the moment, and will soon carry over hopefully into human research, if we program these cells and get them to respond to blue light, and in response to blue light, if we can get them to behave in the same way that rods or cones would behave, we can essentially allow people to see. And just thought I'd show you another image. So the initial research for this was happening back in the time of around 2009, 2010. And this image here is a really interesting one. It's showing a light. It's directly interacting with the neuronal tissue of the brain. Now, a little bit philosophical, a little bit metaphysical, but I think at a conference like this it's really important to consider, because oftentimes, I know not all of us here are clinicians, not all of us are doctors, and a lot of us don't have the intention to be, but I just want to leave you with the thought of some of the bioethical considerations, the moral questions surrounding what it would mean to be enhanced. So inevitably, if this technology is being used for blindness, and we know that the eye is a neural tissue, we know that the brain is a neural tissue, and we're already seeing that you can modify the action of neurons within the brain to behave in a way that we could never even imagine. What are the implications of this? So it's a very high stakes thing. And I think I'll leave it on that note because it's a really interesting thing to think about, and I'd like to thank you all once again for making it here. I really appreciate your time.